Our protocol provides alternative methods for the identification and characterization of viral, nucleolar, and non-nucleolar host factors that maintain the HIV-1 infectious cycle. The concepts used in this approach are applicable to other viral and disease models requiring the characterization of understudied pathways. Troubleshooting techniques are described for modification to other protein models. Demonstrating the procedure will be Haitang Li and Pritsana Chomchan, scientists in the Molecular and Cellular Biology Department at the Beckman Research Institute at the City of Hope, and Roger Moore, members of the City of Hope Proteomics Core in the Department of Molecular Immunology.
Begin by growing an HLFB stably expressing HeLa cell line culture in 3 100 mm culture plates to a 2 × 10⁶ cells per milliliter density. When the cells reach the appropriate concentration, mix 20 micrograms of plasmid containing the Rev nucleolar localization signal 3' flag mutation of interest with 1.76 milliliters of TE7910 in a 15 milliliter tube, followed by the addition of 240 microliters of 2 molar calcium chloride with thorough mixing. Next, transfer the contents of the tube dropwise into a new 15 milliliter tube containing 2 milliliters of 2X-HBS, followed by mixing on a vortex machine. After 30 minutes at room temperature, vortex the precipitation. Add 1 milliliter of the suspension dropwise to each of the 3 100 mm HLFB culture plates while gently swirling the medium. After a 6-hour incubation in the cell culture incubator, replace the transfection mixture with 10 milliliters of fresh culture medium